Welcome, dear students. This is Dr. Hani Nadim, and this is the first lecture on the development of the cardiovascular system. In this lecture, we will cover the early events taking place during development of the heart. Number two, we will list the embryonic sources of the heart. Number three, the steps of formation of cardiac loop. Number four, we will explain some of the anomalies that occur in the position of the heart. When does the heart start to develop? It's the earliest system to develop in the body. It takes place, starts in the middle of the third week. Why is this? First of all, because the amount of yolk in the yolk sac becomes reduced, so there is no sufficient nutrition for the embryo. In addition, as the embryo grows in size, diffusion of nutrients is not sufficient to maintain its growth. Therefore, it has to develop transporting channels to carry the nutrition throughout the expanding body of the embryo. We have to say that the heart starts beating during day 21 or 22 by the end of the third week. By that time, you have to notice that the woman does not even know that she is pregnant. Spontaneous contraction of the heart starts at that time and continues till the end of life. Spontaneous beating is not just a function of the heart, it's a function of isolated cardiomyocytes when they are grown in culture. They spontaneously beat and unite together to form many hearts that synchronize their beating rate. Which layer does the heart come from, or the cardiovascular system come from? It comes totally from the mesoderm. Therefore, we have to revise the development of the mesoderm. This will come next. But for now, we will have to say that blood vessels appear earlier than the heart. This is a picture of the embryo after folding. The first thing to appear are red spots outside of the embryo and inside the embryonic disc. These are made of angiogenic cell clusters, which are called blood islands. They appear in the yolk sac and in the umbilical cord and inside the embryonic disc. Cavitation of these clusters occurs, making endothelial channels, which become elongated, unite with each other to form a network of blood vessels. Surrounding mesoderm makes the muscle layer of these blood vessels. Extraembryonic vessels appear first. This is the vital line vein coming from the yolk sac, going into the body of the embryo. And these are the umbilical vessels coming from the placenta and also going to the inside of the embryo. Extraembryonic vessels appear first, and then later intraembryonic vessels also appear. Two dorsal aortae develop in the dorsal aspect of the embryo. They unite together to form one single dorsal aorta in the caudal part. And drainage of the body is the embryo. A vein develops, which is called common cardinal vein. It's made by union of anterior cardinal and posterior cardinal veins. The anterior cardinal drains the cranial part of the embryo, and the posterior cardinal drains the caudal part. Later on, the heart develops and establishes communication with these vessels, and circulation goes on. As I said, we have to revise the development of the intraembryonic mesoderm. This is a top view of the embryonic disc. As you can see, it's oval in shape, but broad cranially and narrow caudally. It's made of two layers, an epiblast on top and hypoblast below. In the epiblast, a primitive streak appears with a primitive node on its cranial end. From this node, a solid cord of cells grows cranially between the epiblast and hypoblast here in the middle. This is the notochord. From the primitive streak and the notochord, mesoderm develops. Now it's taking the color pink here. Here we are. The mesoderm is absent in 3 sites. This is the site of the oral membrane or the buccopharyngeal membrane. Caudally here we have the cloacal membrane or the anal membrane. And where the notochord is present, this area is devoid of mesoderm. In the buccal membrane and in the cloacal membrane, the ectoderm, the epiblast becomes ectoderm, and the hypoblast is replaced by endoderm. The ectoderm and endoderm are in contact with each other with no mesoderm in between. This mesoderm becomes differentiated. On each side of the midline, it is divided into 3 parts. Paraxial mesoderm, which is divided into somites, followed by intermediate cell mass or nephrogenic cord, which gives the kidneys and genital system. Lateral to this is the lateral plate mesoderm, in which a cavity develops called the intraembryonic coelom, dividing it into somatic layer and visceral layer. The intraembryonic coelom is U-shaped, as you can see here. It has 2 vertical parts and a horizontal part. The vertical parts form the peritoneum and the pleura on each side, of course, and the horizontal part will give the pericardium. Just cranial to the pericardium, the early diaphragm appears. It's called the septum transversum. And just ventral to the pericardial sac, from the mesoderm, the cardiogenic plate appears. From this pericardial sac and cardiogenic plate, the heart develops. There are 3 mesodermal sources of the heart. As we just said, the pericardial sac, which is the horizontal part of the intraembryonic coelom, the cardiogenic plate, which lies ventral to the pericardial sac and develops from the mesoderm, under control of the underlying endoderm, which produces growth factors that stimulate cardiac genes here in this area, in the cardiogenic plate. Angiogenic cell clusters develop. And they develop cavities. They are called blood islands. They develop cavities inside them to make endothelial channels, which fuse together, giving 2 endocardial heart tubes, left and right. The 2 endocardial heart tubes were at first separate from one another, but they fuse together in the middle part, starting here cranially and proceeding caudally. They remain separate from each other in the cranial end and the caudal end. This cranial end, we call it arterial end, and this caudal end, we call it venous end. Just between the 2 endocardial heart tubes and the pericardial sac, a 3rd layer from the mesoderm is formed. This is called myoepicardial mantle. Myoepicardial means that it will give rise to the myocardium and epicardium. Mantle means coat. This is because the endocardial heart tubes will be pushed inside the pericardial sac, having this mantle spread around them to make the myocardium surrounding them. Just between the endocardial heart tubes and the myoepicardial mantle, some mesoderm remains, forming what's called cardiac jelly. As you can see from the names, they refer to the 3 layers of the wall of the heart. The pericardial sac we mentioned here will give the pericardium on the outside. the endocardial heart tubes will give the lining endocardium, and the myoepicardial mantle will give the myocardium and the epicardium. Now let's come to see the changes which occur to these structures as the embryonic disc folds on itself to form a tubular embryo. This is the top view we have seen before. If we have a longitudinal section in it, it will appear like this. Here we have, this is the septum transversum, all right? This is the pericardial sac in the mesoderm. This is the endocardial heart tube, ventral to it. And this is the septum transversum cranially. And this is the buccopharyngeal membrane caudally. Here it is. And this is the forebrain more caudal. You can see that the arrangement of the body structure is not logical. The top part of the body is the septum transversum, or the diaphragm, followed by the heart. Imagine that you have the diaphragm on top of your head, and then the heart, and then the mouth, and then the brain. Okay? What happens corrects this abnormal organization. What happens is folding of the embryo. We have a head fold and a tail fold. And this is what it looks like after folding. Okay? As the head fold takes place here, the organization of structures or the order is changed. The most cranial structure becomes the forebrain, followed by the buccopharyngeal membrane or the mouth. Of course, this is surrounded by the face. And then this is followed by the heart, all right? And this is followed by the diaphragm. We have to notice that this is the head and this is the neck region, right? The heart and the diaphragm develop in the neck. And as the neck continues to develop, both the heart and the diaphragm will move caudally. They will migrate caudally. As folding takes place, the relative position of the pericardial sac and endocardial heart tube becomes reversed. Here the pericardial sac is dorsal to the tube. After folding, the pericardial sac becomes ventral to the endocardial heart tube, which is dorsal to it. Now let's come to see how this endocardial heart tube and pericardial sac will form the early heart, which is called the cardiac loop. 5 changes take place here. First of all, this endocardial heart tube, which was outside the pericardial sac, will invade the pericardial sac. It will be pushed inside it as you push the fist of your hand inside a balloon. So this shows that the pericardial sac will come to surround the endocardial heart tube, forming visceral layer, parietal layer, and pericardial cavity. Okay? If we have a cut section here, we will notice that behind this invading heart tube, there is a double layered fold dorsal to it, which is called dorsal mesocardium. This dorsal mesocardium later on disappears as its cells will die by apoptosis. Disappearance of the dorsal mesocardium will create a cavity behind the endocardial heart tube. Here, this is the endocardial heart tube. This is the cavity, which is called transverse sinus of pericardium. You heard the name transverse sinus of pericardium in the anatomy of the heart. This lies behind the 2 arteries, the aorta and ascending aorta, between them and the 2 atria, the right and left atria. This is the arterial end of the endocardial heart tube, and this is its venous end. The venous end will lead to atria, and the arterial end will lead to arteries. So as the endocardial heart tube is bent to become U-shaped, this transverse sinus of pericardium will come to be reduced gradually in size until it becomes a slit between the two arteries and the two atria. Now, this single endocardial heart tube will have to make chambers of the heart. Dilatations occur separated from each other by constrictions, creating 5 chambers in this single endocardial heart tube. From caudal to cranial, these chambers carry these names. The first one is sinus venosus. The second one is common atrium, one single atrium, and followed by single ventricle. This is followed by a bulbous cordis, and this is followed by a truncus arteriosus. Well, some names are familiar, like atrium and ventricle, but we know that we have 2 atria and 2 ventricles. These will become separated by septa, which will develop in them. All right, so we have one common atrium, it will be divided into 2. One common ventricle, we will end up with 2 ventricles. 3 names are not present in the adult heart. The sinus venosus, all right, from its name, venosus, it gives veins. The major veins, the vena cava system, that will end up in the atrium, and it will give also part of the right atrium. This bulbous cordis, or the cardiac bulb, this will be incorporated into the ventricles. It will give the smooth parts of the ventricles. And this truncus arteriosus, from its name, it will be changed to something else. Arteriosus means that it will give rise to arteries. The ascending aorta and the pulmonary trunk. As the cardiac loop elongates, notice that it becomes longer, while the pericardial sac does not grow to the same extent. So it will not be straight, it will become bent. At first, it makes a U-shaped loop, and later on, as the sinus venosus, which was outside the pericardium, enters into the pericardial sac, it will become S-shaped, as you can see here. The fourth change in the cardiac loop is that it twists to the right. This is a front view. We used to have these chambers in line, one after the other. Now here we will have this is the atrium. Okay, the sinus venosus is hidden behind it. This is the ventricle. The bulbous cordis is twisted to the right of the side of the ventricle. It goes to the right side of the common ventricle. And this is the truncus arteriosus. As the atria grow horizontally, they will bulge on each side of the truncus arteriosus. Therefore, on each side of the ascending aorta and pulmonary trunk, we see bulging parts of the atria, which are the auricles. So, to summarize the story, these are 4 changes. Well, the 5th change is migration of this heart from the neck to the thorax. To summarize the story, we said that the first change is invasion of the pericardial sac by the endocardial tube, making the dorsal mesocardium, which disappears to form the transverse sinus of pericardium. The second change is development of chambers. 2 common names, the atrium and ventricle. 3 strange names, the sinus venosus, bulbous cordis, truncus arteriosus. Elongation and bending, at first U, then S-shaped loop. Twisting, so that the bulbous cordis comes to the right side of the ventricle. Horizontal elongation of the atria, and they bulge on each side of the truncus arteriosus. And finally, caudal migration. Now, as I just said earlier, the heart tube has two ends, a venous end, caudally, and the cranial end, which is called arterial end. The venous end will establish communication with three veins that we mentioned earlier that they develop before the heart. And as the heart tube is united in the middle and separate in both caudal and the cranial end, the caudal end is divided into two horns of the sinus venosus, a right horn and a left horn. Each horn receives those three veins. Number one, the vitelline vein, draining the yolk sac. Number two, the umbilical vein, draining the placenta, or bringing oxygenated blood from the placenta. And the common cardinal vein, which drains the body of the embryo. In addition, the arterial end, which is the cranial end, the truncus arteriosus establishes communication with what's called ventral aortic sac, a blood vessel, that divides into two horns, a left horn and a right horn. Each horn will be connected with the dorsal aorta by 6 aortic arches, one on each of the branchial or pharyngeal arches that develop in the neck. So instead of having one aorta that makes an arch and then descending aorta, in the adult we have ascending aorta, arch of aorta, descending aorta, but in the embryo we have two ventral aorta, 6 aortic arches on each side, and the two dorsal aortae that unite into one single dorsal aorta. Here is a picture showing them. These are the two ventral aortae. These are the 6 aortic arches on each side, and the two dorsal aortae uniting to form one single dorsal aorta. Now we will come to the last section of this lecture. We'll be talking about positional anomalies of the heart. Generally, congenital heart defects have high incidence. Per 1,000 born children, there are 6 or 8 having congenital heart defects. Typical causes are genetic and environmental. The genetic causes are common in Turner syndrome and Down syndrome. You know, Turner syndrome is a female lacking one X chromosome, and Down syndrome is trisomy in chromosome number 21. The environmental causes, the most common cause is infection with German measles virus. The two abnormal positions of the heart that may be encountered are dextrocardia, in which the heart position is reversed. The apex is pointing to the left side, as seen here in the X-ray. As you can see, this is an X-ray of an adult. Probably this person did not know that he or she is having dextrocardia until making this X-ray. It may be an isolated anomaly, or maybe a part of a total reversal of all organs, which is called situs inversus totalis. In dextrocardia, the patient may not suffer any manifestations or complications. The cause of dextrocardia is abnormal twisting of the endocardial heart tube. As I said earlier, the bulbous cordis comes to lie on the right side of the common ventricle. If the rotation is reversed to the opposite side, that is to say, the bulbous cordis goes to the left side of the common ventricle, this will lead to this anomaly, which is dextrocardia. The second anomaly is called ectopia cordis. In such case, we see this poor baby is having a bulging heart inside the pericardium, beating outside. There is no skin here, there is no sternum here. The problem here is not in the heart, but in the development of the sternum. The sternum is made of two sternal plates that fuse together in the midline. If these two sternal plates are not formed, or if they don't fuse in the midline, this part of the anterior chest wall is very weak, through which the heart will bulge. So in summary, in this lecture, we talked about the early formation during the 3rd week of extraembryonic vessels, followed by intraembryonic vessels, followed by development of heart, which starts beating in the end of the 3rd week. And we said that we have 3 mesodermal sources for the heart. All of them are in the cranial mesoderm, pericardial sac, endocardial heart tubes, and myoepicardial mantle. Each gives 1 layer of the heart. Then we said that the endocardial heart tubes will make the cardiac loop through 5 changes or steps. Number 1, the endocardial heart tube invades the pericardial sac, creating a dorsal mesocardium, which becomes absorbed to form the transverse sinus. Then it becomes subdivided into 5 chambers: atrium, ventricle, and 3 different names: sinus venosus, bulbus cordis, truncus arteriosus. Okay, and then it becomes long, making a U-shaped loop followed by S-shaped loop, and then it twists so that the bulbus cordis comes to the right side of the common ventricle, and then it migrates caudally to the thorax. And then we explain the 2 cardiac anomalies, that is to say, dextrocardia and ectopia cordis. Thank you for listening, and see you again in other lectures.